Amino acid adapted metal and metals oxides. Abstract. The smart and multifunctional use of emerging material having novel properties in a green way is the demands of today's life. The quest of new material has fueled the material science in a great extent. The selection of proper material, synthesis, analysis, modification in properties and application are critical on the platform of environment-friendly. Traditionally, inorganic materials were adopted in connection with synthesis, analysis, and application. But intervening of organic material or biomolecules in the inorganic material is playing the pivot role in properties and hence has scope in the application context. Biomolecules like amino acids are available on Earth in different forms. Amino acids are the integral part of life on Earth as they are significant, often called as building blocks of proteins. They can use for structure directing agents or tailoring the properties for the suitable modification of morphology in the specific application purpose. This biocomposite is eco-friendly too. So here in the present review paper we have shed a light on the synthesis methods at different pH, temperature, binding of amino acid to the host and application on metal and semiconductor oxide and calcite modified with various amino acids. Keywords: amino acids, metals, oxidation, biomolecules, nanoparticles, biomineralization. Introduction. Metal and metal oxide nanoparticles. Metal and metal oxide nanoparticles, NPS, have achieved significant position in the various fields of sciences including physics, chemistry, pharmaceutical and biology, etc. The advancement in research of material science has made available the new and versatile materials with suitable activity and properties. We can tune size of the particles which alters the properties of the nanoparticles. These nanoparticles have successfully revolutionized in the sector of energy sector 2.1. Gold nanoparticles are used in drug delivery in cancer cells 2 and biosensing 3. The modification and modulation of crystal properties by proteins are common phenomena in living organisms. The regulation of hard tissues in teeth, bones, mollusk shells are natural examples of such type. The behavior of inorganic nanomaterials in response to biomolecules is tedious because of complex nature of biomolecule. Therefore it is necessary to consider a simple representative system for this interaction. So we have selected only metal, semiconductor, and calcite as inorganic material and amino acid as a biomolecule. Metal 4,5 and semiconductor nanoparticles 6 are showing excellent applications in electronic devices, photocatalyst, sensors, antibacterial and diagnostic applications. TiO2 nanoparticles are major constituents in sunscreen lotion as it absorbs UV light. 7. ZNO nanoparticles have wide applications in piezoelectric 8 and optoelectronic 9 applications. Metal nanoparticles like gold and silver, semiconductor nanoparticles like ZNO, TiO2, CuO and minerals like calcite, calcium carbonate, has unique applications in this context. In the recent era of technology, these metal and semiconductor nanoparticles are building blocks of small electronic components. Therefore the use of nanomaterials in most of the field has created new topic of research about influence of nanomaterials on environment ecosystem. Scientists are indulged to make these nanoparticles biocompatible and functional by use of biomolecules like polymers or amino acid or surfactants like CTAB and PVP, etc. The uses of biomolecules or surfactants are responsible for the variation of shape and size of nanomaterials in this regard. 10, 11. The use of polymer also acts as surfactant of the ZNO host material. 12, 13. The interaction of these inorganic nanoparticles with the biomolecules is governed by pH, charge, temperature, size, shape, and type of biomolecules at the interface. Maintaining the nanosize and also the colloidal stability is extremely challenging. The major challenge can be tackled through the use of suitable capping or doping agents. Some of the amino acids, AA, namely histidine, serine, cysteine, etc., are suitable chelators for metal-based nanoparticles like zinc 14 and cadmium 15. Amino acids are having tendency to modify the morphology of nanoparticles. 16. Since amino acids are naturally occurring biomolecule with abundance in the root zone of the plat, rhizosphere. They are comprising of 1, amino group, NH2, 2, carboxyl group, COOH, and 3, side chain, R, group. Thus, amino acids have 3 functional groups which can react with inorganic nanoparticles. In this manner, interfacings of organic and inorganic compounds are having cardinal importance in order to understand biosensors, biomedical implants or other organometallic compounds. The interlinkage of organic with inorganic compound has found in nature, like in zinc finger, in which amino acid is coupled with ZNO. The representative assemble of organic and inorganic compound in living organism is biomineralization, in which metal ion is linked with biomolecule. 17. Amino acids are building blocks of proteins, enzymes, and muscle tissue which are required to all animals for proper functioning of body and allied physiological processes. The simplicity of interaction of biomolecules with inorganic material has benefits. 1. Amino acids can influence the precise control on shape, size, crystallinity, or other chemical property of inorganic materials. 2. This reaction takes place at low temperature with organic solvent. 3. Diverse types of amino acid are available for modification of morphology and capping enable to alter physical property of inorganic material. 18. On an account of above discussion, we have considered metal binding properties of amino acid for some metals nanoparticles like O, Ag and semiconductor nanoparticles like TiO2, ZnO and calcite. There are 20 natural amino acids, so diverse metal binding alternatives are available. There are several amino acids which are used as reducing and capping agents in the metal nanoparticles synthesis. 19, 20, 21. Gold and silver nanoparticles. Gold and silver nanoparticles are tremendous application in the biomedical field like bioimaging. 22, targeted drug delivery. 23 and biosensing. 24. The use of gold nanoparticles in biomedical field is owing to the unique surface plasmon resonance, SPR, property of gold nanoparticles which can be tuned by size of nanoparticles. The activity of proteins in the formation gold nanoparticles has studied. 25. Amino acid interfaced gold nanoparticles. From Roman times gold nanoparticles were used to stain glasses with beautiful shades of colors. Faraday in 1857 reported colloidal gold nanoparticles. 4. By considering the implementation of gold nanoparticles in biomedical field which needs benign functionalization instead of harsh chemicals and biocompatible. Amino acid can be used as a reducing and capping agent for metals, semiconductors, and insulator. 26, 27. Simulation study of binding preferences on gold nanoparticles were analyzed. 28,29. The unique linking between nanoparticles with amino acid is governed by pH of the solution. At acidic, low, pH nanoparticles linked by a amine group, near to carboxylic acid group COOH, while at high pH due to repulsion between O surfaces and negatively charged A carboxylic group this bonding is not takes place. 30. The detailed investigation between gold nanoparticles with amine group of amino acid has been studied. They proposed the interaction of amino acid and O nanoparticles via amine group and cross-linking of O colloids via hydrogen bonding in thiols containing O groups. 31. Amine stabilized gold nanoparticles were also analyzed. 32. Tryptophan has been act as a reducing agent. 33. The inherent property of gold nanoparticles to bind and form complex with amino acid can be used to detect amino acid if proper functionalized. Quercetin functionalized gold nanoparticles are capable of visual detection of amino acid like arginine, histidine, and lysine. 34. L-cysteine molecules in gold colloidal solution are interlinked through sulfur, NH3+ and Cu groups. 35. The adsorption of histidine on gold nanoparticles were analyzed by soft X-ray photoelectron spectroscopy and near-edge X-ray absorption fine structure spectroscopy. Vacuum deposition of thin histidine films on polycrystalline gold was inspected by high-resolution synchrotron photoemission. 4 monolayer films amino groups, N3 atoms of imidazole rings, and carboxylate groups are probably involved in the bonding to the gold. In the multilayer film histidine form 3D aggregates held with strong intermolecular forces in between them and weakly interact with gold substrate. The equal proportion of protonate amino group and protonate imidazole are existed there. The soft X-ray for multilayer film induces neutral molecules in a mass loss via decarboxylation. 36. Within vacuum chamber the cysteine molecules were evaporated on standard gold 111 single crystal substrate. When this deposited material is analyzed by scanning tunneling microscopy under ultra-high vacuum conditions, it was observed that at room temperature and sub-monolayer deposition elongated molecular islands having no molecular order with a highly ordered molecular layer of quadratic symmetry are coexist. Upon increasing dosage of deposition structures with short-range hexagonal order were observed. After annealing to 380 K drastic change in surface morphology and adsorption structure with superstructure characteristic for alkane thiol self-assembled monolayers on O, 111, was observed. It was concluded that covalent SO bonding takes place by the deposition. 37. First principles quantum mechanical treatment, density functional theory, was performed on interaction of tryptophan gold interaction. 38. It was found that carboxyl and indole functional groups were involved in the interaction with metal. It depicts p interactions along with small ionic interactions instead of van der Waals interactions between tryptophan and O nanoparticles. Tryptophano complex has shown fluorescence quenching in tryptophan by introducing hybrid states associated with O as low-lying excited states which gives non-radiative energy transfer to these states. Gold nanoparticles were synthesized by assisting various amino acids. 39. The details of synthesized method for gold nanoparticles assisted with various amino and pH are given in following table. Amino acid interfaced silver nanoparticles. Silver nanoparticles are excellent class of material having antibacterial properties 50, 51. Therefore, they are preferred to use in the decoration of sweets by thin foil, food packaging. The stability of colloidal solution was found higher in basic pH 52. The interaction of different amino acids like cysteine, tyrosine, protein, glycine, alanine, serine, methionine, cysteine, lysine, arginine, aspartic acid, tryptophan with silver ions has been explored. 53. Silver nanoparticles are capable of detecting amino acid like cysteine and cysteine which has sulfur in their backbone. 54. Silver nanoparticles are used for colorimetric sensing of cysteine. 55. Cysteine is a thiol-containing amino acid which has critical importance in rheumatoid arthritis, Parkinson's disease, and heart disease can be detected by silver nanoparticles. 56. The biomimetic synthesis of silver nanoparticles has possible by taking advantages of silver-binding peptides. 57. The adsorption of biothiols onto the surface of AN has given new strategy for analysis of biothiols and sulfur drugs. 58. Biothiols are adsorbed on the surface of AGNP by forming a covalent bond. Cysteine and glutathione has ability to form metal ligand cluster. 59. Amino acid like cysteine has tendency to modify the metal surface, silver, in such a way that cysteine silver nanoparticles are dispersed in water. 60. They also showed that the aggregation of cysteine-capped colloidal silver particles is interconnected by hydrogen bonds. The hydrogen bond was observed to form between amine and carboxylic acid functional group. Amino acid like glycine, neutral nonpolar, L-tyrosine, neutral polar, L-aspartic acid, acidic polar, and L-lysine, basic polar, coated silver nanoparticles exhibit stable and homogeneously dispersed nature. L-tyrosine coated silver nanoparticles have highest catalytic activity for methylene blue reduction. 61. Glutamine, GLN, and histidine, His, functionalized silver nanoparticles are used as colorimetric sensor for Hg2+ in water because it provides amino, NH2, and carboxylic acid, COOH, binding sites for Hg2+. 68. Histidine modified silver nanoparticles manifests less cytotoxicity and has application in cancer therapy. 69. Lysine functionalized silver nanoparticles are used for detection of histidine and histidine-tagged proteins. 70. L-histidine functionalized silver nanoparticles indicates that histidine is adsorbed on the NP surface through the imidazole moiety with the Cu functional group projecting outwards. 71. The complex of silver and tryptophan are showing pronounced antibacterial activity while the complex of silver and histidine are showing lower antibacterial activity. 72. The silver zeolite shows that the antibacterial activity has inhibited by the addition of amino acids like L-cysteine, L-methionine, L-histidine, L-tryptophan. 73. According to group, 74 histidine has adsorbed on silver nanoparticles surface through carboxylate group while through imidazole ring on gold nanoparticles. By the same group from the DFT calculation it was interpreted that the coordination via the carboxylate group was much more stable on Ag than on O by 17.4 kcal/mol. It has reported that glutathione stabilized silver nanoparticles contain SH group can acts as a colorimetric sensor to probe Ni2+ in water. 75. Serine acts as a reducing agent in synthesis of silver nanoparticles by a microwave irradiation. 76. These nanoparticles are observed to stable for more than 6 months. Basic amino acid like L-lysine or L-arginine is excellent reducing agent for uniform, water-soluble silver nanoparticles. 77. At pH 7 from surface enhanced Raman spectroscopy L-arginine links with Ag surface through the guanidinium fragment while at pH 9, L-arginine links with Ag surface through guanidinium, carboxylate, and the aliphatic moieties. 78. Glutamic acid capped silver nanoparticles were synthesized from silver nitrate, glutamic acid, and glucose by a chemical reduction method. 79. The presence of glutamic acid screen nanoparticles from agglomeration. The capped nanoparticles make them biocompatible too. Cysteine has tendency to adsorb on metal surface by 3 functional groups, NH2/NH3, COOH/Cu or SH/S. Cysteine biomolecules were used as electrode modifier 80, 81, 82. L-cysteine molecules in silver colloidal solution are attached through amino and carboxylate groups. 83. Immobilized silver nanoparticles are more stable, showing less aggregation ability with less oxidation in aqueous medium as compared to colloidal silver nanoparticles. Arginine-modified silver nanoparticles exhibits immobilization of silver nanoparticles on ZNO nanorods. It also displays enhanced antibacterial activity for E. coli and Bacillus subtilis bacterial cells and no cytotoxic effect on human. 84. Amino acid interfaced Cooper nanoparticles. Histidine interacts with the Cu. 110 surface only via the carboxylate group and ring imino, N2, nitrogen observed in the XPS study and annealing at 425K. The XPS analysis indicates strong interaction of IM ring with the Cu surface by means of imino nitrogen 85. Reflection absorption infrared spectroscopy, RARES, study confirmed that adsorption proceeds via the carboxylate group, Cu, and via the dehydrogenated nitrogen of the imidazole group 86. According to this group there are 3 binding sites for adsorption of histidine on metal surface, an amino group, carboxylate group, and imidazole nitrogen atoms. On gold, histidine mainly adsorbs through the depronate carboxylate group in a quasi-upright geometry that maintains the imidazole ring far from the surface. On copper, the histidine NH2 and Cu groups are involved in the interaction with the surface, with the cycle closer to the surface. 87. X-ray absorption and emission spectra study and cluster models under DFT theory has proposed that glycine molecule at 400K formed binding with both oxygen and nitrogen atom to Cu nanoparticles. 88. Screen-printed carbon electrode modified by copper nanoparticles can used for efficient detection of all 20 amino acids. 89. This method of detection of amino acid is more stable than other traditional methods like Raman, NMR, or circular dichroism. Amino acid chelated copper nanoparticles indicates enhanced antibacterial activity against Enterococcus faecalis as compared to copper nanoparticles. 90. It was investigated that copper nanoparticles modified carbon paste electrode can employed for detection of L-arginine and L-lysine. 91. In aqueous solution the PN of imidazole ring of histidine was able to form stable complexes with Cu, 2, Ni, 2, and Zn, 2, 92. Barlow et al. Has systematically explained adsorption of glycine on Cu, 110, and Cu, 100, takes place below 290K. While it bonds through oxygen atom of carboxyl and nitrogen atom of amino group below 408K. They have studied alanine adsorbed on Cu, 110, S-norvaline on Cu, 110, S-methionine on Cu, 110, S-serine on Cu, 110, and S-proline on Cu, 110, also 93. 1. Semiconductor nanoparticles. The binding and nature of interaction of biomolecules with inorganic surfaces are significant for biological evolution theories. 94, biomaterial implants. 95 and biosensors. 96. I. Amino acid interface TiO2 nanoparticles. Titanium dioxide is one of the most studied and useful material in variety of fields like paints, sensors, catalyst, and electronics devices. The interaction of biomolecule with TiO2 important in the view of understanding the reaction with implanted titanium metal. Surface spectroscopy clears that glycine was adsorbed on TiO2 surface, 110, through carboxylate group 97. The surface modified with glycine when inspected under scanning tunneling microscopy which evident that glycine has been dissociatively adsorbed on TiO2 surface. 98. The adsorption of glycine, methionine, serine, and cysteine on rutile, 100, and 110, surfaces were studied by simulated performed with first principles molecular dynamics. Serine forms bond between a deprotonated hydroxyl and Ti surface while cysteine forms bond between thiol group and Ti surface. 99. The detailed analysis of valence band spectra, VBS, and XPS of glycine adsorbed on TiO2, 110, surface at room temperature has revealed that layers formed by glycine molecule is in polar zwitterionic form. 100. They have investigated that multi-layers of glycine adsorbed on TiO2, 110, surface via zwitterionic form through 3 peaks in VBS at 1, 18.1 eV attributed to C2s from a carbon atom, 2, 27.6 eV attributed to O2s from carbonyl group 3, 24.5 eV attributed to N2s from NH3+, amino, group. Glycine layers get desorbed when they are faced by photons and remaining layer has C, NH1, NH2, O and Ti3+ surface species. In connection with this DFT approach was used to investigate the contribution of van der Waals forces in the interaction of glycine with the TO2, 110, surface 101. It has proved that dissociative adsorption is more favorable. They have found that van der Waals forces do not affect surface deprotonation. DFT approach was used to get insight of adsorption of proline and glycine on the TiO2, 110, surface 102. As per this approach, the most stable configuration is anionic moiety connecting two surface titanium atoms via the carboxyl oxygen atoms. Amino acid forms hydrogen bonds with the surface. Owing to amphoteric nature of the TiO2A and zwitterionic nature of amino acid, the carboxylic group replaces surface O group on Ti site and connects to Ti103. They have analyzed XPS of adsorbed amino acid on Ti surface and observed that amino acids are adsorbed at only acidic milieu and no adsorption in basic pH. The XPS study of adsorbed amino acid on Ti surface has concluded that adsorption is pH dependent due to amphoteric nature of the TiO2 104. Thermal desorption spectroscopy of glycine adsorbed on TiO2 surface has observed that glycine layer doesn't desorb at room temperature 105. The temperature-programmed desorption, TPD, investigation of surface chemistry and binding of DL-proline on a TiO2 single crystal, 001, surface has been carried out 106. Adsorption of proline leads to creation of surface defects. The core level spectra of C1s lies at 288.0 eV, which is much lower than non-dissociative adsorbed carboxylic acid for proline adsorbed on TO2 110 single crystal surface. In addition to this, the peaks corresponding to CH2 and CNH has also found. The core level spectra of N1s shows peak corresponding to NHX species due to CNH, 400 eV, and other corresponding to CNH2+, 401.5 eV. The O1 spectra corresponding to carboxylate group is clearly seen at 532.5 eV. The presence of oxygen defects at the surface zwitterionic proline as less oxygen sites available to facilitate the required proton migration from the hydroxyl group of the amino acid 107. Adsorption of glycine is not observed on rutile surface of TiO2 because of amino group of glycine forms hydrogen bind to water in solution, which weakens the binding to TO2. This principle is applied to polar solvents like CH3CN and CH3O. Interaction of organic matter is derived by pH and is insignificant at pH 7. At pH 9 the hydroxylation is drastically reduced and deprotonation of the carboxylic groups occurs 108. XPS and temperature-programmed desorption, TPD. Techniques are used to probe the adsorption of glycine on rutile TiO2, 011, single crystal. The peaks of Ti, 2p, or O, 1s, was observed to attenuate by adsorption. Glycine was observed to form zwitterionic, H3 plus NCH2 with less stability in the dissociated, NH2CH2 structure with more stability on TiO2 surface 109. Glycine adsorbed on the TiO2 surfaces like 110, 100, and 001, were monitored by glycine-modified AFM tips, atomic force microscopy, probe. It is clear that glycine showed a desorption peak on the TiO2 surfaces and not on mica and pyrite. TiO2, 001, and 100, surfaces show same adhesion forces and shape of the force curve. Whereas in 110, surface the orientation may influenced by glycine adsorption 110. The interaction of glycine with the rutile TiO2, 110, surface was examined by high-resolution core-level photoemission and scanned energy mode photoelectron diffraction, PhD, of the O1s and N1s. There is affirmation of zwitterion's nature NH3 plus CH2 with multi-layer deposition, at low coverage only deprotonated glycinate species, NH2CH2 is present. Multiple scattering simulations of the O1's PhD data convey that glycinate is bonded to the surface through the two carboxylate O atoms. The hydrogen atom from deprotonation form hydroxyl species. Non-appearance of major PhD modulations of the N1's emission is consistent with the amino N atom not being involved in the surface bonding 111. The first principal calculations were employed to study the adsorption of proline and glycine on anatase, 001, surface. The calculated adsorption energy confirms that amino acids strongly interact with 001 surface in the dissociative adsorption manner. This interaction in between oxygen atoms of the amino acid and surface titanium atoms is responsible for binding of amino acid to TiO2 surface. The dissociative adsorption configuration for proline is having maximum interaction energy of -1.81 and -2.38 eV, while for glycine maximum interaction energy is -1.53 and -2.11 eV. 112. Surface oxygen to form more hydrogen bonds with the hydroxyl hydrogen of the biomolecule, therefore L-serine adsorbed on anatase TO2, 101. Surface forms biocompatible substrate in benthic microbial fuel cells 113. In the dynamic study of histidine adsorbed in TiO2 nanoparticles by attenuated total reflectance. Fourier transform infrared, ATRFTIR, spectroscopy has ascertained that both the imidazole side chain and the amine group interacting with the TiO2 surface and the adsorption to be reversible. 114. Photodegradation study of L-Alanine adsorbed on TiO2 was performed. This adsorption is observed to possible when L-Alanine is zwitterionic and titania surface is positively charged. The palette of this material was kept under UV light source through quartz wall, then it is outgassed followed by recording IR spectrum. It was observed that photodegradation possible through removal of protonate amino group followed by mineralization of carbon atoms due to O radicals. 115. The amino acids like serine containing O group, histidine, and tryptophan containing NH and asparagine containing NH2 in their side chain appeared to adsorb more favorably by the side chain. They are vulnerable to photocatalytic oxidation and showed high decomposition rates. However, alanine having aliphatic side chain interacts with the acidic bridged O groups on TiO2. 116. Theoretical study of adsorption of cysteine on TiO2, 110, has been carried out and it was concluded that cysteine interact with TiO2, 110, surface through carboxylic group, the amino and sulfur groups. 117. The systematic X-ray photoelectron spectroscopy investigation was employed in order to understand that mechanism of bonding off L-cysteine molecules on a rutyl TiO2, 110, single crystal surface at -65 degrees Celsius and room temperature. It was clarified that deprotonated carboxylic group forms a double bond with Ti atoms. Thiol groups remain intact with Ti atoms. 118. The infrared studies have observed that when pH of the solution varies in range 2 to 8, cysteine is adsorbed on TiO2 from aqueous solutions. At pH 2.0, cysteine is adsorbed mainly in protonate and zwitterionic forms. At pH 5.0, cysteine is adsorbed in zwitterionic forms. At pH 8.0, the electrostatic interactions between protonate amine parts and negatively charged TiO2 surface are favored. 119. 2. Amino acid interfaced ZNO nanoparticles. Organic reaction at oxide surfaces has well illustrated for ZNO, TiO2 and other oxide surfaces. 120. With relatively cheaper and biocompatible ZNO has always been benevolent material for application perspectives. The bio-inspired approach when applied to biomolecules like amino acid and inorganic phase nanostructures, then the examples of such types are very obvious in nature. Some natural examples are histidine is connected to zinc and zinc finger, configuration of DNA binding protein. 121, guanine crystals found in the skin of many fish. 122 and many zinc complexes with amino acid. 123. The zinc salts when mineralized from zinc nitrate and hexamethylene tetramine, HMTA, in presence of amino acid, then variety of morphologies are formed as per type of amino acid. 124. The presence of citrulline, GLYCYL-glutamine, and proline led to form micron zincite crystal, histidine form nano-sized crystals, arginine from nanoscale platelets, GLYCYL-glutamine form generates ribbon-like coating. Histidine prevents the tendency of ZNO to form larger and elongated crystals. Thus, amino acids molecules are acting as structure-directing agent. The systematic explanation of formation of histidine-capped ZNO has observed by considering the ratio of Zn2+ to O and Zn2+ to histidine molecule. 125. They have observed that histidine or O are binding to Zn2+ at different constant values and therefore Zn, O, N and Zn, C6H9N3O2, N complexes are formed. The variety of morphologies like prism, flower, and crystalline hollow microsphere were formed with the presence of L-histidine by varying the ratio of zinc ion and hydroxide ion. 125. This group has proposed that at the lower concentration of hydroxide ion, histidine molecule is competitive to hydroxide ion to coordinate to Zn2+. As a result of this flower-like structure is formed. At the higher concentration of hydroxide ion, Zn, O, N complexes are formed, which is responsible to form anisotropic prism-like structures. The XPS analysis of nitrogen, carbon, and oxygen atoms revealed that alanine was found to adsorb on the nonpolar surface of ZNO, 100, in zwitterionic form. 132. It confirms that carboxylic acid group is linked to ZNO surface. Glutamic acid acts as surface modification agent in ethanol, which generates the variation in morphology like twinned cone, twinned prism. With glycine twin prism structure with smooth surface, sharp prism, and symmetrical sections. 133. Aspartic acid, glutamic acid, and alanine are efficient surfactant for ZNO results to various structures. 134. Addition of histidine in ZNO suspension inhibits the antifungal activity against C. albicans, as histidine is a quencher of hydroxyl radicals and singlet oxygen. 135. The antibacterial effect of ZNO was observed to decrease by the introduction of histidine for Escherichia coli bacteria 136. Thus quenching effect is evident by above observation. The influence of ZNO nanoparticles, size 50 nm, on the environment in terms of the reaction between four representative amino acids, glycine, L-glutamic, L-aspartic and L-histidine, with ZNO nanoparticles has been screened. The increase in the concentration of amino acid has resulted in decreased in particles sized distribution, decreased in percentage zeta potential and reduction in agglomeration 137. Prism-like and flower-like morphology of ZNO and indium-doped ZNO is obtained by hydrothermal synthesis method with the aid of histidine 138. Anastasia Briff et al. Have incorporated various amino acids in ZNO crystal by simple chemical method. They have found that incorporation of different types of amino acid in the crystal induces strain, alters band gap and change in the variation of morphology. Serine and arginine were observed to incorporate to highest level and linear correlation between lattice distortion and band gap alteration has been observed. In connection with this they have found that when amino acid modified ZNO is annealed at 300 C for 90 minutes will decompose the amino acid results to strain and band gap relaxed 127. Substantial influence on morphology of ZNO has been found by the presence of gelatin in aqueous solution 139. From the study of binding constant and number of binding sites with thermodynamic parameters and the nature of binding forces confirms that in the complex of L-tryptophan ZNO nanoparticles the binding forces are van der Waals interaction and hydrogen bonding 140. Density functional theory, DFT, demonstrates that arginine and aspartic acid has good affinity to interact with nonpolar surface of ZNO than histidine 141. The harsh chemical condition and high temperature requirement can avoid by using environmentally friendly material are preferred to use for synthesis. ZNO synthesized by using amine and citrate additives 142. The tuning of band gap of ZNO can achieve by using histidine acts as capping agents for ZNO nanoparticles 143. ZNO nanoparticles of 20 nm and 70 nm treated with L-serine were showing no agglomeration with any alteration of surface charge 144. Coating of citrate or L-serine to powder of ZNO nanoparticles induces negative or positive surface charge respectively 145. The presence of amino acid during the synthesis of ZNO nanoparticles will tailor the band gap via inducing the strain the host crystal 146. ZNO synthesized in presence of glutamine shows rose-like morphology, in presence of histidine shows dandelion-like morphology and in presence of glycine shows flower-like morphology. Glycine has simpler structure having only 2 carbon atoms leads to loosely assembled structure with larger size. Histidine has complex structure having more number of carbon atoms leads to close-packed structure with smaller size. 147. The lysine molecules are used as shape-directing agent in hydrothermal method. It generates dumbbells, twin prisms, whiskers, and bundles-like morphology for different concentration of lysine. Lysine-decorated ZNO exhibits good photocatalytic activity for decomposition of NO under UV vis light irradiation 148. The unique nest-like ZNO hierarchically porous structures were obtained with the aid of precursors zinc acetate, glycine, polyvinyl pyrrolidone, PVP, and sodium sulfate 149. This porous nature has high sensitivity and fast response and recovery speed suggesting potential applications in gas sensing property. For ZNO core shell nanoparticles L-cysteine molecules are serving as a source of sulfur for ZNS shell around ZNO 150. This makes ZNO nanoparticles more stable in water by cysteine molecules and results in enhancement in zeta potential. Photoluminescence indicates a significant blue shift of 35 nm in near band edge emission with decrease in defect luminescence. The existence of cysteine makes this material biocompatible also. CO precipitation method is employed for synthesis of cysteine-capped ZNO/graphene oxide composite. The synthesized composite manifests good photocatalytic activity for degradation of di-RHB 151. The systematic exploration of control in size of ZNO was achieved by using cysteine molecule with zinc nitrate and HMTA. The pH was kept to 6.3. It produces the hollow amorphous ZNO microspheres 152. Cysteine functionalized ZNO quantum dots depicts optical absorption in the visible range and good colloidal stability over wide pH range, pH 2 to 12. It was interpreted that chemical coordination of the ligands to Zn2+ centers, which concomitantly reduce the numerical density of oxygen defects on or near the surface 153. Biomolecules like L-lysine, L-cysteine, and L-arginine can engage to control the shape and ultimately properties of ZNO. The synthesis was carried out with zinc acetate, 0.5 mol of amino acid in a 100 ml solution of distilled water ethanol, 1:1V/V. The amino acid assisted ZNO in conjunction with multi-walled carbon nanotube are used as a glucose sensor 154. Pure ZN substrate was dipped in solution, pH 10, containing ZN2+ and L-cysteine generates 1D vertically aligned ZNO nanorod array 155. The steady, time-resolved NFTIR spectroscopy was employed to investigate the specific binding of L-tryptophan with nanoparticles of ZNO. Results derived from both experiments and theoretical computations demonstrate that the ZNO nanoparticles form H-bonding with NH group of amino acid, but when both COOH and NH are present in amino acid, then ZNO nanoparticles prefer to bind the COOH group rather than the NH group through H-bonding. The effect of H-bonding is strong in neutral medium, pH 7, while it is weak in basic, pH 10, and acidic, pH 4, medium 156. Ag/ZNO nanocomposites were synthesized by Solve-Othermol method in presence of L-arginine. This nanocomposite manifests excellent photocatalytic activity for methyl orange 157. L-arginine modified ZNO indicates enhanced nonlinear refraction and nonlinear absorption with blue shift in excitonic absorption peaks 158. Biodegradable nanocomposite synthesized from ZNO, starch, and lysine can be useful in food packing applications to improve the shelf-life of the food products 159. Paper-like ZNO nanowire films when treated by L-lysine can enhance the UV photoconduction and suppress the oxygen vacancy-related photoluminescence 160. ZNO flowers with nanorods have been synthesized by using lysine as capping agent 161. Molecular dynamics simulations approach was employed to study the interaction of amino acid and xeno-water interaction. Glycine exhibits the strongest attraction to 1010, with binding energy 2.57 kJ/mol 162. The largest binding energy 7 kJ/mol was found for tyrosine interacting with the surface in which the ZN ions are at the top. The surface with ZN atoms at the top has binding for all amino acid with 2.60 kJ/mol, whereas the surface with O atoms at the top has non-binding for most amino acid. Self-consistent charge density functional based tight binding, SCCDFTB, method was performed on 1010 ZNO surfaces for glycine adsorption 163. Glycine molecule binds to the 1010 surface through both functional groups for 0.5 and 0.25 monolayer coverage. Glycine molecule binds to the 1010 surface through the medium of carboxyl or the amine group for 1 monolayer coverage. Electronic properties of the surface get affected when adsorption occurs through amine group. In all cases the glycine adsorbs dissociatively for all coverages. Density functional theory clarify that arginine and lysine are the preferential binding sequence for ZNO surface with the larger adhesion energy. Arginine is the amino acid with the strongest adhesion on the dry surface, while lysine is the most stable on the hydrated surface 164. The adsorption of glycine on zinc-terminated polar 0001 surface has been studied by density functional theory 165. A study at low coverage glycine adsorbs on ZNO surface preferentially in anionic form with carboxylate moiety forms a ZNO co-ZNO with ZNN bond between amine and ZN surface. The detailed study at high coverage revealed that a glycinate bilayer is formed at the ZN-ZNO surface. First layer of glycine adopts honeycomb ZNO lattice. In second layer ZN glycinate complex are observed in which ZN ions are chelated by glycine through the carboxylic and amine group. Density functional theory calculations were performed to study site-specific interactions between tryptophan and ZNO for tryptophan-ZNO nanoparticles complex. The calculations demonstrate that COOH group is more actively participate in the bridging than indole and amine groups in tryptophan. The interactions between ZNO and tryptophan were facilitated by ionic and hydrogen bond. These complex exhibits quenching of fluorescence due to non-radiative energy transfer by pp* of tryptophan indole functional group 166. 3. Amino acid interfaced calcite mineral. Calcite occurred in building material in marine organisms. Synthetically it acquires a regular rhombohedra structure along with fracture by cleavage along low-energy lattice planes. 167. Calcite, CaCO3, is naturally hierarchical materials and one of the naturally inspired crystalline materials. It existed in the three forms aragonite, minerals calcite, and vaterite. Depending on the charge, side chain, or nature of amino acid the crystal gets modified. 168. Cysteine, histidine, and arginine have a profound inhibition on calcite crystal growth when these amino acids are bound to crystal 169. This induces the surface covered with the holes. Further, it was observed that the formation of aragonite on crystal substrate was taking place around the holes produced by inhibition. Owing to different charge and polarity of side chains at applied experimental conditions, L-aspartic acid, L-lysine, L-asparagine, L-tyrosine, L-phenylalanine, L-serine and L-alanine were used for the interaction of this amino acid with calcite crystal at pH 10.76 170. The nonpolar amino acid has low consequence on structure, morphology, whereas it is more pronounced bipolar amino acid. L-aspartic acid has most engineered structure, morphology with strong distortion on C direction. Low concentration of L-aspartic acid leads to formation of vaterite, while at higher concentrations only calcite has been observed. In case of L-lysine, L-asparagine, L-tyrosine, and L-serine, the formation of additional steps on surfaces are seen. Impact of organic additives like aspartic acid and glycine on amorphous calcite crystal has performed. End 171. It was shown that the increase in concentration of amino acid the lifetime of crystal. The most influence was observed for aspartic acid, and it shows strong binding to calcite. Therefore crystal shows rounded edges with aspartic acid and sharp edges with glycine. The interaction of all amino acid with calcite was explored, which demonstrated that cysteine energetically interacts with calcite 172. They have observed that tryptophan has induced maximum distortion followed by phenylalanine in calcite lattice. Controlling crystal structure and morphology of calcite in synthetic material has been observed 173. The amino acid interacts with the calcite in an isotropic manner. Coherence length of amino acid mediated calcite crystal is lower than calcite crystal without amino acid, determined from the interpretation of high-resolution synchrotron X-ray radiation 174. The effect of binding amino acids on interfacial energies of growing crystal of calcite has investigated from atomic force microscope, molecular modeling studies, X-ray photoelectron spectroscopy and surface X-ray diffraction studies 175. The unprecedented finding of increase in hardness with the incorporation of amino acid in calcite crystal was observed 176. Conclusions. The remarkable alterations in the properties of the inorganic matrix material were observed when biomolecules are capped, embedded, adsorbed, incorporated, functionalized, or modeled in it. The binding of amino acid to the host matrix are governed by the temperature, pH, and type of amino acid. The simple synthesis method, biocompatibility, and performance of these organic-inorganic biocomposite are the profound property. The broad spectrum of technological applications in various fields requires proper knowledge of how amino acids attached and detached to inorganic matrix with different parameters. Biomineralization is an approach by which inorganic material is produced in some species. This paves the way to search new engineering between organic and inorganic material to meet new demands in medical and technological field. The substantial efforts of theoretical and practical approach to investigate and understand the details of the optical and electrical properties of these bio-nanocomposites are incredible for the future application in the various fields. These engineered new superior materials have opened a window for the tremendous applications in diverse sectors like biomonitored or biocontrolled air purifier and biofilter.